I can work with this. Miosis, the division of gametes into more and more gametes. How does it work? Well, questions like these will be answered and let me just interface. You got the growth, you got the DNA synthesis, you got the growth. What we're actually going to start off with is drawing some chromosomes, some good old chromosomes, you know, the things that house the genes and all that. 4 male chromosomes. Now, what this is going to do is that it's going to allow our cells to give ourselves some traits, if you will, some good old fun. And next, we're going to draw the female chromosomes, the mother chromosomes. This is a red marker. This is a red marker. So we're just going to draw some mother chromosomes here in the middle. We need to draw the alleles in the chromosomes. They are the thing that make the chromosome a chromosome. So, long chromosome has more alleles than a smaller chromosome. We just drew some lines on some other lines. That ain't a chromosome. What we actually need to do is that we need to label these alleles. We're just gonna put a lowercase a here and a lowercase b here. Yeah, no. You see here, I put a little tricky on you. That's a capital A, not a lowercase a. Now, what does this mean? Why did I do that?
Lowercase a, capital A.
It means heterozygous, when they actually combine. It'll come into play later when things actually matter. Homozygous, dominant and recessive. Now, homozygous dominant means that all of these things have uppercase stuff. Homozygous recessive is kind of like this one, where it's just, you know, basic. Now we have our complete set of chromosomes. Now that we got our alleles and all that down, we essentially completed G1. The first step of the process, this took 20 minutes to do, is we have to multiply, the synthesis phase. See this chromosome? Now there's one of it. They're no longer one strand. These are not X chromosomes, they are X-shaped chromosomes. Now there's G2. Edit this. Prophase. Here's the female chromosomes and here's the male chromosomes. They've crossed over. They've done the allele thing that I mentioned before. Editor, mention me saying the thing from before. Is that it's going to allow our cell, once it divides, to give ourselves some traits, if you will. And that's pretty good. Metaphase. We have aligned all of the previous chromosomes into one line. Attach these chromosomes to spindle fibrils. Now what this is going to prepare to do is that it's going to split apart like these 2 chromosomes. These chromosomes have the same alleles. It's going to be used to split up the cell later, which I will explain now. Anaphase. Anaphase is what metaphase tried to do. Metaphase, it created spindle fibers. Anaphase actually uses them. They pull apart the homologous pairs to prepare for telophase. Here we are, the main event. Telophase is where everything starts to frickin' shatter. This is the nuclear envelope. 4 chromosomes. This single cell has 2 nuclei. Look at these stretch marks. It really wants to split apart. This one cell is gonna split up into 2 cells. 3, 2, 1, bam! It's done it. 1 cell, no more. This baby is 2 cells now. Can you frickin' count? This is cytokinesis right here. Now you have half of the amount of chromosomes we had before, because now we have 2 cells that completely start over.